Welcome to solverfy.
Hello students, the first step in designing an experiment to study the expression of homologue of the EVE gene in cranefly embryos is to identify the sequence of EVE gene in the genome of the cranefly embryo. Once the sequence is known, then the next step would be to design primers for the gene and use them to perform PCR to amplify the genome or gene from the complementary DNA or RNA isolated from the cranefly embryos. Now the amplified DNA that can be cloned into a suitable expression vector such as plasmid and transformed into bacterial or insect cells. The transformed cells that can then use to express the EVE protein, which can be used to purify and then used to raise antibodies against that protein. The next step would be to use these antibodies to detect the expression pattern of the EVE gene in cranefly embryos. One approach would be to perform immunohistochemistry on cranefly embryos at different stages of development using the antibodies to detect the presence of EVE protein in different tissues and cells. This would allow us to determine the spatiotemporal expression pattern of the gene during the embryonic development. Another approach would be perform in situ hybridization to detect the expression of the EVE gene at the mRNA level in cranefly embryos. This would involve designing probes complementary to the mRNA sequence of the EVE gene and hybridizing them to tissue sections of cranefly embryos. The hybridized probes can be then detected using a suitable detection method such as fluorescence or enzyme-based methods. Ultimately, the data obtained from these experiments would provide insights into the role of the homologue of EVE gene in cranefly embryonic development and the conservation of HOX gene function across the different organisms. I hope this video is useful for you. Thank you.